afternoon, everyone. I'm here to talk about two different types of microsampling devices, the volumetric absorptive microsampling device and capillary microsampling, and the application of these to clinical studies. Initially, I'll talk to you a bit about study design and how the things to consider when using microsampling in clinical studies, and then I'll walk through our experiences with these microsamples. As Sam mentioned, I lead our clinical microsampling group. Our group is based and supported by the Burns Trauma Critical Care Research Centre and the CRE Reduce at the University of Queensland. The aim of our research is to reduce the volume of clinical sampling, that is, to reduce the burden of blood taking by removing just a very small specimen of sample and then using these to reliably measure drug concentrations. So this is a brief video of the wicking of blood onto the tip of a VAMS device. As you can see, the blood moves onto a polymeric absorptive tip, and on completion of filling, you have 10 microlitres that is being collected. This is then available, after drying, this is then available for storage and transport to a bioanalytical facility for mass spec analysis. The other type of microsample is the capillary microsample, which Chris talked to us about, and where we're using this, we are using a plastic variety. What you can see is we collect our sample into a fine capillary tube which contains lithium heparin. This is commonly done in the Children's Hospital locally, and this sample can then be centrifuged, the red blood cells removed, and we can collect then a liquid plasma sample. We can apply it to filter paper to prepare dried plasma spots, or we can apply it to a VAMS device to prepare a plasma VAMS. To implement microsampling techniques into a clinical environment, there are some stages to think of. There's the study design, where we consider the factors which may impact on our study, and we test these in the experimental phases, which is our bioanalytical validation and clinical bridging study. We perform bioanalytical validation, which is prescribed by the FDA and EMA. They supply us with good detailed testing for our normal clinical samples, where we do traditional sampling and we have a liquid plasma sample. The FDA have also recently provided information for dried blood spot analysis, and we can use this as a bit of a guide of the information or the sort of testing we need to perform when doing our other types of microsamples. The type of testing that you're looking at when you're using a microsample is hematocrit testing, dried sample testing, and also if some people take a capillary tube and dilute it prior to storage or transport, and then you need to do a dilution integrity test. Our clinical bridging study is where you compare a microsampling technique to the traditional gold standard of plasma sampling in patients. And this is also an opportunity to test arteriovenous sampling to peripheral sampling. We currently have a clinical bridging study underway at the Royal Brisbane Hospital in the ICU, and we're testing some microsamples in adults prior to moving them into a paediatric study. So for study design that incorporates microsampling, the decisions that you make are based on multiple factors, and we offer a contingency approach to managing this process. We go into detail in this in a paper we published in 2016. This contingency process is based on thinking about the properties of the drug, the characteristics of your patient, and the location of your patient, and the patient's proximity to the bioanalytical facility. So I'm just going to consider an example, and I'll run this example through my talk, is where we've got a clinical pharmacokinetics study of vancomycin in critically ill paediatric patients in South Africa. It might be in a remote hospital where our resources are limited, but we'll conduct our bioanalysis outside South Africa, so say Brisbane. And what we need to think about is the study feasible in using microsampling? First thing to think about is our drug. What do we know about vancomycin and its binding? So this is whether or not it's going to be sitting in red blood cells or in plasma. And what we know about vancomycin is that it's almost exclusively found in plasma. We've tested this in the laboratory. What that means is if we're collecting whole blood, the red blood cells serve to dilute our sample. And we have to think about our sensitivity and whether or not that dilution effect is going to impact on our results. And we perform this in our bioanalytical validation. For stability, we know that vancomycin is extremely stable in plasma, frozen, but we need to look at how it is in whole blood, but also if we're going to use a dried sample, the stability in as a dried sample. Again, we do this in our bioanalytical laboratory validation. We then need to think about the suitability of our sample. Are total or unbound concentrations required? And are we going to have profound patient group variability that's going to affect our results? A study in 2017 by de Kock on critically ill paediatric patients found the unbound concentration ranged from 49 to 98%. So one of the big questions for us is, can we measure unbound concentrations of vancomycin? I will discuss this a little bit further. The next question then is, will the site of sampling affect our result? And so we need to know, if we peripherally sample, will we be able to compare this to our central result? And this we correlate in our clinical bridging study. We also have to consider the location of our sample, and our patient is remote from our analysis. So dried format microsampling is ideal, and that would help for transport and storage. Again, we need to test our stability in a laboratory validation. And finally, there's our patient. We know they're critically ill, so they're likely to have a low or variable hematocrit. The impact, therefore, of the variability of hematocrit on a microsample needs to be considered. And again, we take this into our bioanalytical validation. Okay, so this is a bit of a description of our adult study that's occurring at the Royal Brisbane Hospital at the moment. Here we have our patients receive a dose of vancomycin, and then for each time point, we collect 5 different samples. We collect a peripheral sample of whole blood VAMS. We collect a capillary peripheral sample, which we centrifuge and apply the plasma to a VAMS device. We're then collecting an arterial sample, so this is 3 to 4 mils of whole blood, more our traditional sample, and we apply this to whole blood VAMS. We then centrifuge this sample, take the plasma, and apply this to a VAMS, but we also freeze it, and this is our liquid sample. So to compare these results and start to see the feasibility of these microsamples, we need to develop and validate 3 different types of methods. We need a liquid plasma bioanalytical validation. We need vancomycin in whole blood on VAMS, and we need vancomycin in plasma on VAMS. So we first started with a liquid plasma assay, and we recently published vancomycin total levels using 10 microlitres of sample. We also measured urine and renal replacement therapy effluent. You can see our validation data. Our acceptance criteria is 15%, and our data here met those requirements. You'll note here we measured unbound concentrations, but we did this with 200 microlitres of sample, which is the volume we need to use at this stage to measure unbound concentrations. So, going back to our patient, this is our traditional sample, where our gold standard sampling of plasma, where we collected our 3 to 4 mils from an arterial line. We did use our liquid sample assay of just 10 microlitres, and this is what our plasma result is for our critically ill patient. Our next sample was the whole blood on VAMS. So we ran through a validation here, and I'll run through the results quickly. We found we had a recovery of 69%. This you don't need, there's no acceptance criteria for recovery, it just needs to be reproducible. We didn't have a measurable matrix effect. Our intraday precision and accuracy was within 10%. We tested a variety of patients at the lower limit of quantification, and we had less than 4% precision. We tested a range of hematocrit from 19 to 59%, which should cover a good range of patients, and we found our results were within 10%. We also tested stability, so extreme temperature stability, 5 days at 50 degrees, and our deviation was within 2%. We kept our samples at room temperature for 106 days, and again, deviation within 5%, and when stored in the fridge or freezer for 58 days, less than 5% deviation. We also tested drying time across 22 hours. So we applied the sample to a VAMS and we watched it across 22 hours to see how it dried. For this, we found a deviation of 5%. At the time, we also tested dried plasma spots, and we published our finding, which was that we found a recovery effect across actually 56 hours, and it deviated 30 to 40%. This meant for downstream testing that if you dry your clinical samples for a different length of time to your calibration standards or QCs, your concentrations for your clinical samples may be underrepresented or potentially overrepresented depending on the length of time. It was at this stage that we moved away from dry plasma spots for vancomycin. And we also checked our ratio of red blood cells to plasma and found it was indeed in the plasma layer. So when we look back to our patient and we assay their samples, what we find is that our whole blood VAMS from our arterial sampling is in green, and our whole blood VAMS from our peripheral sample is in grey. And you can see they're significantly lower than our plasma sample. But what we're comparing here is plasma to whole blood, and you'll recall our red blood cells are serving to dilute our sample. So when we apply a correction factor to just measure the concentration now in the plasma, but also our standard curve is prepared in a different hematocrit level to our patient who had low hematocrit. When we apply this correction factor, what you can see is that our whole blood VAMS from the arterial line does indeed provide results that are quite similar to our plasma concentration. We did do a simple regression where we check our plasma concentration to our whole blood concentration, our whole blood concentration from the arterial line, and we get a slope of 1, which sounds great, and an R squared above 0.9 something or other. But we're very cautious in this sort of approach in analysing our data. I can get a slope of 1 quite comfortably with very variable data, so we prefer not to use this sort of method of analysis. The FDA recommend the use of not specifically for microsampling, but for our studies they use a test called an incurred sample reanalysis. We perform this in all of our clinical studies. It's where you take 7% of your samples, you reassay them, and you compare the original result to the reassay result, and you look at the deviation to the mean, and if it's within 20% for a small molecule or 30% for a large molecule, it's acceptable. This data did meet that acceptance criteria. You'll see the peripheral data is lower, certainly with the Cmax, and this may be useful clinically if there's a distribution effect of vancomycin. It's hard to draw any conclusions. It's a single patient. We're waiting to see more data. When we measure all of the peripheral concentrations against our arterial, we've got a 67% at the peripheral the peripheral concentration is 67% of the arterial line, but we've got a variation of 20%, so we're just going to have to wait and see. But you recall from our study design, we have more peripheral data from this patient taken at the same time points. So to now look at our plasma data, this is our bioanalytical validation of now vancomycin in plasma on VAMS. Now we've got a recovery of 105%. Again, no measurable matrix effects. Our intraday precision and accuracy comfortably within 10%. Our selectivity at 6.3% at our LLOQ across a variety of patients. And our stability testing, we tested here 47 hours at 50 degrees, so that might be stuck on the tarmac at the airport. Room temperature, fridge and freezer, all for 51 days, very little deviation in sample. And here is our drying time across 56 hours, and our deviation was 10%. So when we look to our patient profile, what we see is our plasma VAMS from our arterial line consistently overestimated the central plasma concentration. But again, it was within the acceptance criteria of our incurred sample reanalysis. And here's our patient peripheral data. This time, with plasma acquired as VAMS, the peripheral concentrations are 76% of the arterial plasma concentrations. And you'll recall when we used whole blood, it was 67%. The precision here was 30%. So it's just too variable to really draw a conclusion at this stage. So the key points of where we are so far is our bioanalytical validation meets our acceptance criteria, that is for liquid vancomycin in liquid plasma, which we can do down to 10 microliters, which means we can do it from a capillary sample. We can also do vancomycin in whole blood on VAMS and vancomycin in plasma on VAMS. For our bridging studies, we've got promising results. They're very early. We definitely need more patient data. It will give us information on our peripheral versus arteriovenous sampling. It'll also give us information on our capillary versus liquid plasma sampling. We're wary about where we go because we do know that to dose vancomycin in critically ill children effectively, we need data on unbound concentrations. The same study by de Kock just recently suggested that we can correlate protein concentrations to our unbound levels. They provide us with a calculation to achieve it. And so I guess one of the questions is whether or not we can obtain this information of a protein concentration as part of our routine clinical practice. So where are we with our example? For whole blood VAMS, we know that if that's to be successful, we're going to need hematocrit levels. We're probably going to need protein or albumin concentrations for our sample. We also still need to see our peripheral versus arteriovenous sampling. If we're in South Africa, and I was hoping we'd have a South African intensivist here, but no. But anyway, whether or not that's available as routine clinical care, I don't know. If the answer is yes, then I think it's still feasible. It is still a possibility that that could happen. If the answer is no and we can't get hematocrit, albumin or protein, then the next option really seems to me at this stage to be a liquid plasma collected by capillary. Still need to see peripheral versus arteriovenous, but to get that sample, we need a hematocrit centrifuge. So then we did actually have the hematocrit, but we can, then we've got the possibility of whether or not we can use that small sample, or possibly pooled samples, to also measure an unbound concentration. So the example that I used highlights that there's There's no, the importance of study design prior to experimenting and performing these other parts of the study. There isn't a one-size-fits-all approach from microsampling from what we can see, and this certainly doesn't appear to be a perfect microsample. It's just going to be what's going to be the best microsample for your patient, depending on what location they're in. If we're to successfully use microsampling in pharmacokinetic studies, we think the best approach is probably a really thorough and considered study design from the start. Okay. Thank you.